Like DNA, RNA strands can also pair to form a double helix. The paired nucleotide bases are packed in the middle of an RNA helix, surrounded by the backbone. The RNA helix has a different geometry than a standard DNA helix. For example, the RNA helix has a hole down the middle and also has a significantly narrower and deeper major groove. The backbone is composed of repeating ribose sugars and phosphate groups. Unlike the 2' deoxyribose used in DNA, ribose has a hydroxyl group attached to the 2' carbon. This extra hydroxyl group influences the secondary structure and is the primary reason why an RNA helix has a different shape. Base pairing between strands is similar to that in DNA, except that adenine pairs with uracil instead of with thymine. Uracil is never used in DNA. As in DNA, a GC base pair in an RNA helix has 3 hydrogen bonds. The AU pair, like the AT pair, has 2 hydrogen bonds. RNA strands often fold into complex structures. In this stem loop structure, also called an RNA hairpin, a single-stranded RNA molecule folds back onto itself. The stem at the bottom is a classical RNA helix. Bases in the single-stranded loop, in contrast, may be unpaired or engaged in non-standard interactions.